actually three housekeeping things. One is that, just to tell you what's coming up, because this is the end of my presentation, but next 2 weeks, it'll be David Elkins. Now, for you who have been going to these Nobel series for several years, you'll recognize that David has done the economics and the Peace Prize every year, I think, for about 4 years. And Valmai Elkins has done the Literature Prize. And both are excellent. So that's what's coming up. The next thing is that you may be aware, I got it in my cell phone today, that, where are we here? I don't want to listen. Where are we? Neighbor and North Family Health Team. I don't know whether you saw that. Any of you members of the Niagara Family Health Team? Well, I mentioned that because there was kind of an alert there about emergency rooms and intensive care units overloaded again, and part of it's COVID. I don't think it's the flu yet, but there may be a few cases of that, and certainly with the kids, it's this respiratory viral, this RSV infection, and I don't see any reason why the latter shouldn't affect adults as well, but it's largely a pediatric problem for now. Anyway, so it clearly is an issue. So maybe I would ask you next week to wear a mask in here. Or if you want to today, it's hard for me to talk with a mask on. But I think it's a good thing. You know, it happens with this now, what, the third fall going into the winter when, I mean, it's amazing how, with the people being indoors and people not wanting to distance, not wanting to wear masks, do those things, and then it really hits. And I'm very aware of that because my wife is in a long-term care home, but I'm also aware that a lot of other people have symptomatic disease in those 3 years. And it kind of came up in the fall, American Thanksgiving, not a good thing. And then right into Christmas, right through New Year's, and then it really hit. And so I just mentioned that. Upcoming, I mean, it's been lovely to have it face-to-face. And then the other thing is that when I was putting this together this morning, I thought about my father. Well, my father was a chemical engineer, the first in his family to go to university. He was a graduate of Cornell. And Dad was a quiet, very, very well educated, broadly educated person. And, you know, we went fly fishing for smallmouth bass in the summer and the evenings, and sometimes the weekends in the upper Thames and Aiken rivers. But we didn't talk a lot about stuff, and we certainly didn't talk about chemistry and his work. But I'm very conscious of that now because after doing this, the 6th year of the Nobel series, there have been a series of Nobel Prizes given in chemistry, and they all fit together, like we're talking about click chemistry today. They all fit together, they all click together. And that can't be accident on the part of the nominating, the people who make the nominations and those who finally decide who win the prizes. Chemistry's obviously made big progress. So I thought, well, and then I ended up writing a piece about this, but I think I'd better get on with things, and then if we have time, I might deal with that later. But it's an interesting kind of story. Well, it's always nice to start off with something that at least a little perspective. These are 2 galaxies, as I'm sure most of you have surmised. They're probably about the size of the Milky Way, so 100 billion stars in them, around that. And they're very close. I mean, this is what's going to happen. If this was the Milky Way, this will be Andromeda, except that Andromeda is much larger than our Milky Way, believe it or not. And within 4 billion years, they're going to merge. That doesn't mean they're going to hit each other. I mean, there's a hell of a lot of space between stars. So they may just kind of do this. But certainly their black holes will merge. The giant black holes that were highlighted in the physics prize in, 2020, at the center of the Milky Way. And they will have a very large black hole at their center. But you can see that already there's a gravitational effect, because you see some of the gas and some of the other debris, that kind of stuff, star debris, stardust if you like, being spun off from this one and moving into this galaxy here. And that's kind of what happens with gravity. So here's a nice one. This is the gang of Nobel winners. Put this way, they look like prison pictures, don't they? But all the science ones are at the top. Here, there's Pablo that I talked about the first time with ancient DNA, and these are Valmai's and David's winners. But anyway, there they all are. Not many. It's not many, really, is it? Annually. Now, we're going to talk today about creating complex organic compounds. And this is where my dad would kind of be, his ears would kind of perk up, because this has been an issue in chemistry almost from the beginning, of how do you fit different molecules together, how do you build complex molecules. Well, we're going to see how people have done it here. One thing you might be surprised about, but maybe not when you think about it, nature creates by far the most complex molecules in the universe. And it does so with efficiency and ease. By comparison, star chemistry is far simpler than biological chemistry. And there's been a big threshold here around the year 2000, well, the late part of the 20th century and the early part of this century, a big change in chemistry. And what have been the major changes in chemistry? The advent of, we're going to talk about this in a bit, directed evolution as a way of selecting the best enzymes, click chemistry, which is all about this year's Nobel Prize. And one thing I'm not talking about is artificial intelligence, which is doing a great job of figuring out the shapes of proteins, well beyond what humans can do. So that's the change. Now, AI is shaping a whole lot of other things too. That would be worth talking about sometime. Well, let's look at some of these selectively, the ones that bear on what we're talking about today. My favorite in 2018 was Frances Arnold. She has a really interesting biography. Well, you know, all of the Nobel laureates are asked if they're willing to write a biography, kind of a story. Most of them don't, but 30 or 40% of them do. Arnold's is well worth reading. She's a substantial woman in so many ways, and she's had her share of triumph and tragedy, as many of us in this room might have had too. Anyway, well, let's just see. Well, there they are. There's Frances. In the same year, George Smith. I'm not going to talk about him or Gregory Winter, but Frances did something, in my mind, that was really original. Now, here we are in the 1980s. Well, building molecules is a very tricky business, as you'll hear me repeat 2 or 3 times later. They're really tricky to kind of assemble if you're trying to assemble a protein, amino acid by amino acid. I mean, there are 22 of them, and you can imagine the permutations and combinations possible as you kind of string them out. And some proteins contain hundreds of thousands of amino acids. So you can imagine the changes. And form dictates function, because depending on the order of the amino acids will dictate whether these kind of strings of amino acids kind of turn right or left or twist on themselves or hide certain polar groups or not. So she tried, as many did before her, to rationally design enzymes. Those are things that speed up reactions, and most of them are proteins. RNA is also an enzyme. Not as efficient as a protein, but it's also an enzyme. So she thought about this, and like others, found out it was maybe not a waste of time. You can certainly do it, but very laborious, very costly, and maybe not the way to go. So she shifted to using what she called directed evolution to modify an enzyme called subtilism. And what she did is create random mutations in the enzyme's genetic code. So the gene that dictates that particular enzyme, she introduced random mutations into it. And then she introduced those mutated genes into bacteria. Bacteria are like little factories. If you give them kind of mutant genes, they'll make the corresponding protein product. So what you end up with is a whole bunch of variants of this enzyme. And then she tested each of those variants for how effective the enzyme was. And then she repeated this whole cycle several times. And at least with this particular enzyme, by the third cycle, the enzyme had become 200 times more effective. So she's allowing evolution, and not strictly speaking, she's also playing a part because she's introduced the mutations into it. But still, it's a very remarkable idea, and it worked, and very efficient. So that's Frances. It's one of those great ideas in science. And as far as I know, no one else tried it before she did. So, get ahead of myself here. Now, no surprise with the next one. Here's gene editing, Emmanuel Carpentier and Jennifer Doudna in 2020, two remarkable women, each contributing. The key here was not CRISPR so much as was the great insight that bacteria encounter in their lifetime thousands and thousands of viruses, and they keep log of them, and they copy a part of the DNA or the RNA of the bacteria to their own DNA. And then when an intruder, a viral intruder comes along, they scan their little library. Have we seen this guy before or not? If they have seen that one before, then that becomes the guide molecule, the RNA guide molecule that latches onto the virus, coupled with an enzyme, and destroys it. So it's a great surveillance device in bacteria, but the insight here was to realize, gee, we could take advantage of this to edit any kind of gene if we have a guide. If we know the target gene that we want to change, then we need a guide molecule that recognizes that particular gene. So, in other words, the same DNA sequence for that particular gene, part of it or all of them. So that's what it was all about. It's not the best picture of the two of them. My favorite picture, which I couldn't find for this one, is the two women walking arm by arm up to the podium to get their prize. It's a wonderful picture. They're great. They certainly look a lot better than these pictures. Two very attractive women. I shouldn't say that, but they are. Anyway, so here's the thing, the guide. Here's the guide. It's been, now you can, I've said this before, you can buy this stuff. It's actually fairly cheap. If you can send away to, I don't know, there must be dozens of firms now, and give them the sequence of bases or nucleotides. You tell them what the guide is, they will make it, and they'll send it to you in the mail. And then that guide goes looking for its mate. in a strand of DNA. So, I mean, this is the cartoon version. They kind of pulled a strand out from a chromosome here, stretched it out so you can see it. But here's the guide. It's found its mate right here. But that guide is also linked with an enzyme here, Cas9, but there are Cas8, Cas7s, variants of Cas9s, and other enzymes here, and it has the effect of snipping the DNA, both strands, in the double helix. And if you do that, and you don't do anything more, like here, then nature does a kind of a crappy job of repairing the wound, and it has the effect of silencing whatever that gene is, which is great if you're using this tool for Huntington's chorea. You don't want that gene to work. So that's a great tool. But there is another way. You can add to the package, the backpack, the molecular backpack, the correction, and insert it into the gap. That's the perfect fix. You insert the corrected gene. That works. So it's gone on beyond them. I won't say beyond them, but there are a lot of people using that technique now, and I mean, very, very diverse ways. By the way, here's, you certainly wouldn't recognize this as CRISPR-Cas9, but that's kind of what it looks like in this cartoon version of the stuff, but you don't need to go any further. Well, now, we'll move on to 2021 now with Benjamin List and David McMillan, but we'll stick with List. Because List asked, I mean, a lot of science is asking the right questions. And if you have, say, an enzyme, a protein that functions as an enzyme, do you need the whole thing? Or do you just need this very small part of that protein, that enzyme? Well, List asked that very question. Turns out no one else had asked that question. And it turns out, well, you don't need that whole package. You only need a small part of it. And one of those illustrations, well, there's a picture of List. At least these people are in their 70s and 80s. But they're their pictures at the time of the year. But here's the thing. Here's the whole, here's that enzyme. I mean, this doesn't begin to show how complex the whole thing is. But it turns out that only a very small part of that whole molecule is actually effective that confers that property that you want. And in this case, it turned out to be proline here. And so you do it. So his insight and what he learned by experiment was, yes, you don't need the whole molecule, you only need the active part. Well, that simplifies things, doesn't it, when you're manufacturing enzymes. So, well, let's move along. And the next one is one of my favorites, not because it's any better done than the others, but because at least I was on familiar ground. This Nobel Prize in 2021, there were two of interest, which we'll talk about, but the Nobel Prize in Physiology or Medicine in 2021 was awarded jointly to these two, well, he's the older guy and the younger one, for work done independently by both, David Julius and Ardem Patapoutian, for discoveries of receptors for temperature and touch. Well, I mean, that's part of the neurological examination, assessing sensation and recognizing that there are different sensations for temperature, modest changes in temperature, more extreme temperatures, touch pressure receptors, even itch. Patapoutian, the guy on your right, had a paper in Nature this year for itch receptors. Would you believe it? There are actually receptors, specific receptors, proteins, for itch. I wouldn't have thought that. But anyway, that's what he found in his group. And show you how real interesting these are. This is a touch pressure receptor, which looks like a prop, three-bladed prop on a boat, right? at the back from the top. This is a side view, that's from the top. If you press down on, let's say this is, you're looking down, and you press down on the skin over here, or over here, or over here, what happens is those blades shoot out from one another, opening up a gap in the middle through which ions can go that reduce the transmembrane potential and begins the action potential. It goes all the way from there, all the way up to the spinal cord, and through various connections up to the sensory cortex. So, I mean, these are very elaborate constructions, and I can tell you that right to insects, they're incredibly complex, these receptors. You probably don't know that, I mean, that some insects have, hundreds of thousands of neurons. The ones that we find in our garden. It's hard to believe that little thing right there that might bite you has a lot of cells in it. So anyway, that was great work, and so we'll move on. I'm trying to make space for my little letter at the end, but anyway. Now I said to here's the here we are. Here's this one. Artificial intelligence cracks the code of protein complexes, providing a roadmap for new drug therapies. There's no doubt that AI, and Google was one of the first into this, and then they've had kind of breakaway divisions. I forgot the name of the division that's involved in this, the AI molecular structure, but that's what they've done. And it is incredible how good AI can be at designing proteins and figuring out their. Here's another slide. I mean, AI predicts the shape of nearly every protein. I mean, this is a huge revolution. This was laborious work to figure this. It took years to figure out what the shape and composition of some molecules were. And here's AI figuring it out in a week or days. It's amazing. So, sometimes AI does stupid things. And so here it was asked, this is DALL-E, a program that draws anything. Here it was asked to draw 4 teacups in the shape of an avocado. Well, that doesn't turn me on, but I guess, it's kind of fun. But as you may know from the brain series, AI is now being used to create quite elaborate and what we would think, if we thought that way, very imaginative art. And it's doing the same thing with poetry. And it's writing articles. In fact, there was another article in Nature just last week. Can AI write a science article, given the right kind of prompts? Well, not so well. But it tells you, if this is where you are in 2022, imagine where you're going to be in 2052. It'll almost certainly be capable of writing the article long before that. And now here we are in the group. I could ask Rita here. I think this is this Swedish up here. Here it is in English. There's the Swedish original. And then I looked it up. English is generally shorter than French. But here's a language that's shorter than English. Gets the job done with fewer letters, apparently. Anyway, here's Carolyn Bertozzi, quite an amazing person, which I'll comment on later. Morten Meldal from Denmark. And the wise man, Barry Sharpless. He had won a Nobel Prize in 2001. So this is the second prize. He's obviously the leader in this. Not the best, but the leader. So let's look at, it's always worthwhile, I don't usually highlight this, but look at people and say, where did they come from? Where did they train? It turns out that something like 10 or 15% of universities, 80% of people with tenure in the United States come from something like 15 universities. No surprise, there are differences, right? But here's Carol. I mean, look where she's been. UC Berkeley. I mean, that's in the top 10 in the U.S. And now she's at Stanford University, top 10. And Barry Sharpless, an American, Stanford University, right up there, top 10. He's at the Scripps Research Institute, which is where retiring Nobel Prize winners go. And that's where Crick went, Watson and Crick. And it's hard to evaluate Meldo, except the University of Copenhagen is very good, very classy. So, you know, and that's gets into all kinds of interesting questions about selection for such universities. Now, there they are. I love this little cartoon. So there's Meldo, Bertozzi, and Sharpless clicking molecules together. There's the click spot, click, click. Now, how did they get there? And, well, I'm going to read some of the parts straight from the Nobel, because sometimes in their arcane way of expressing things, that's maybe the best way. So, the Royal Swedish Academy of Sciences has decided to award Carolyn Bertozzi, Morton Meldo, and Barry Sharpless the Nobel Prize for the development of click chemistry and bioorthogonal chemistry. Well, what is bioorthogonal? There's a mouthful for a cocktail party. But it refers to chemical reactions which proceed rapidly and very selectively in biological environments such as the cell without affecting any other chemical reactions in the cell. So they're very specific in what they do. So it's almost like a parallel process going on. I mean, cells are incredibly complex. There are thousands and thousands of proteins, different proteins in cells, and other chemicals or molecules inside cells. So you can imagine if you stuck something in something that would change the metabolism, the metabolic rate inside a cell, it might affect a whole bunch of things. But these are very specific, very designer-specific molecules. And their work was beautiful, elegant, and a compelling story of how the best scientists work by defining the challenge, figuring out how to solve it, and working collegially toward the end. That's a characteristic of modern science. Modern science beginning, say, 1900. Or maybe later than that, maybe 1930, 1940 on, large collaborative science. It's international. They have a shared kind of common language in chemistry and mathematics. And by and large, they get along. It doesn't seem to matter what their nationality is or what their color. Well, in part, it's more of an issue some places than others. But, so, well, now this is strictly for, this is an interesting thing here because, Jane, pay attention. This is the role of ideation and conceptualization, pretty bizarre, conceptualization in science. Modern chemistry is driven not only by important discoveries and improvements, or improvements and new discoveries, but also by the formulation of concepts that catalyze, mean move it ahead, and accelerate new development. Concepts foster innovative thought processes. That's a really interesting statement to make. This is a prefrontal exercise. This is highly cognitive here. That open up novel pathways, you might even argue pathways in the brain, to address different challenges, Both ideation and conceptualization are fundamental important tenets of science and technology. I absolutely agree. Which expand our current knowledge and develop new perspectives. Now, in a more general sense, we can all share the last part here. As humans, we all need occasional, sometimes surprising eye-openers to stimulate our minds and concepts to energize and expand our understanding, you might say, but you could add other things in there. So we all need to be pushed and not nudged from time to time. And the more novel it is, the better. Now, a little bit in the background, I mentioned this earlier, but the late 1900s was a heyday for molecular biology. It certainly was. One Nobel after another, with new discovery after discovery. But there was a big stumbling block. Building, as I said earlier at the start, building complex molecules is costly, time-consuming, and often leaves unwanted byproducts behind. So that's not a good way to go. We dealt with the first line here, but this just described what chemists were kind of like in the nature of their job in the late 20th century. They had become very skilled and talented, and they had learned to master the methods to perfection, and were able to make astonishingly complex structures with high fidelity, and reached a point where they could almost make any molecule they could imagine or find in nature. But the trouble was that the whole process was tedious, time-consuming, expensive, and unique to building a specific molecule. That makes it really expensive in time and money. In a word, it was frustrating. Enter sharpness, the old man of this thing, 2 years younger than I am. In the 1990s, one of the most creative chemists, this is the Nobel Committee saying that, one of the most creative chemists, Barry Sharpless, who had been awarded the Nobel Prize in 2001 for earlier work, had been giving this some thought. Well, think about the thought experiments of theoretical physicists. No, same kind of stuff that's happening here. He recognizes a roadblock or a stumbling block, and it's a huge one, and how to overcome this. He realized that efficiently manufacturing complex molecules would strongly benefit from a more, what he called, streamlined process, where each compound could be made using a limited set of highly robust and efficient reactions. In other words, fewer pieces that could be, as you will learn, clicked together to make the more complex molecule. So rather than relying on a plethora of possible methods, all with their specific scopes, in other words, this individual thing, I'm going to create molecule A, a really complex one, it's going to take me several years to get there. So rather than that approach, how would you speed it up? Well, he presented, interestingly enough, the Nobel Committee actually cited an abstract he had written in 2001. I mean, it's got to be pretty rare where somebody cites an abstract, where he kind of put his thoughts together in about 400 words, 300 or 400 words. And then, very shortly, a few months later, out came the article. The article, Click Chemistry, Diverse Chemical Functions from a Few Good Reactions. That completely changed the field around. Completely changed it. We're talking about moving 180 degrees in the other direction. Described how complex functional molecules could be efficiently made using a modular approach. So, and I mentioned this, the field changed, and he put it in this, in one paragraph here. If you want to build complex molecules, begin with smaller, easier to manage molecules. It's like solving human problems. Could be managing staff. whole bunch of other things. It's kind of a modular way of thinking, isn't it? If you want to build complex molecules, begin with smaller, easier to manage ones, with carbon skeletons, of course, because, yeah, which could be clicked together by employing chemical reactions that form strong bonds that don't interfere with other molecules and leave no unwanted byproducts behind. So it's clean, it's fast, it's efficient, and it's very specific. Okay, that's what he advocated, and guess what? Well, I'm not going to go through all of his stuff again. Move ahead here. Within a year, not bad, working independently, Sharpless and Melville, working independently, discovered what was later likened to the crown jewel of the new field of click chemistry. The jewel was the reaction between an azide molecule, which contains 3 nitrogen atoms, not important to you, and an alkyne molecule, which contains a triple bonded carbon atom, which does matter. The two clicked securely together in the presence of a copper catalyst, much as matching Lego pieces click together in an often used analogy by chemists. Click. If you could hear it, it would be clicked. Snapped together. That means when it snaps together, it's like Lego, it's secure. It's hard to pull apart. And it's very specific. And so here's the cartoon. There's the azide here. Here's this little click thing on both ends. This kind of goes into here, right? In the presence of copper, they actually come together. So there's the click in a cartoon. And there is this IKEA analogy here, which you'll be familiar with. In a broad sense, the principle was like the IKEA kit approach, in which all necessary components, the building blocks, once you take the cardboard sides off, along with a set of easy-to-use hardware, which in this case are the reactions, chemical reactions, were provided, together with reliable instructions. And then the comment made that almost anyone could follow. Well, not almost everyone, not everyone could follow IKEA instructions, but most. Okay. Yeah, I can hear that that's true. Now, we know a lot about DNA, we know a lot about RNA, we know a lot about proteins. What we don't know much about are sugars. Sugars are glycans. They form vast and very large molecules. Sometimes they cover cell surfaces. They link up to proteins, glycoproteins. But we don't know much about them. That's what Bertozzi was really interested in doing. A brand new field, exploiting this whole field that no one knew much about. So she needed a click reaction without the copper catalyst, because copper is toxic to biological systems. And she discovered, it's the way it happens. She has a large lab. She herself went to the lab, or went to the library, and did her work in the stacks, and she found in some obscure corner in some journal that somebody had done exactly that. They changed the alkyne molecule into a ring-like structure. And bingo, she had the answer. The alkyne azide reaction with no need for copper. So no toxicity. So she got around this. That in itself is worth a Nobel Prize. And she kept going. And by the way, that's what this strained version kind of looks like. It's been likened to, what are those things you have around pools, those long things that bend? What's the name of those? Anyway, they require a little force to kind of bend them into a circle, but there's that constant tension in it. So there's that potential energy right there, and as soon as you click it with the other one, it's released. And it's a one-way reaction. You can't go back. Once made, once clicked together, that's it. So that's a good kind of reaction. Now, She also had a bright idea. Well, this is her second bright idea. But it was born of frustration. In the early 1990s, she was beginning her work on glycans, and she tried mapping a glycan whose job apparently was to attract immune cells to lymph nodes. But she found it took her 4 years to get this stuff done. Now you can imagine a bunch of graduate students, postdocs, docs, and other people around there all struggling with it for 4 years. Very frustrating. So she said, I wonder, is there a way around this? And because what she wanted to do with glycans, for which we know so little, or we don't know very much, she wanted to be able to visualize them, see them, be able to track them on cell surfaces and within cells. And she could use that, you could visualize it, if you could somehow attach to the glycan a fluorescing molecule. That when you shine light on it, it kind of emits green or whatever the color happens to be. Kind of a beacon. So it's like attaching a little flashlight at the end of the glycan. But how do you do that? Well, she listened to German science, so she goes to meetings, she pays attention, she's not out drinking coffee or something actually doesn't work. And by the way, that's what her colleagues say about her, and people who work for her now and have worked with her and for her in years back. She's a great mentor. She's very well known for that, as is Sharpless, of a different age a bit, but she's apparently a great mentor to many people. Well, so in that little obscure paper, he mentioned that he could get a variant of sialic acid. That's a form of kind of attenuated form of a sugar. Could the cells be nudged into making it? And if they could be nudged into making it, could be nudged into making not only it, but attach a little handle to it that you could click to, either side of the combination. So and then why not attach a fluorescent molecule to the handle? Well, you can follow the process here. I mean, here's the cell. Starts out, Bertozzi fed the cells with this modified sugar. There's the sugar, there's the azide attached to it. Away it goes in. It actually attaches itself, there it is. This now is attached to the glycan on the surface, but you can't see it. So what she does is she takes the alkyne, the other side of the click equation, and attaches it to a fluorescent green molecule, and then clicks the two together. Now she has a molecule that together she can see in the cell. And there it is, the fluorescing molecule just inside the cell membrane, that green fluorescence. By the way, that's going to be a Nobel Prize for the use of various molecules that fluoresce. It's a great way of tracing all kinds of stuff. And there's another Nobel right there. By attaching a fluorescing molecule to the modified alkyne, which would click with the I'm repeating myself here. She has a tool for tracking sugars, complex sugars, glycans, and glycoproteins inside cell. That's a great tool for any number of studies. And in so doing, revolutionized the whole field in biology and elevated it almost to the level of studies of DNA. That's quite an accomplishment. I think she's the star of the three. And Sharpless had the vision at the start for sure, kind of launched it, made it possible. Maybe it's the two of them, but anyway. Now, coming to, well, one of the things that these glycans on the surface of well, let's bring it right home to coronaviruses. The tip of the coronavirus that connects to the cell is covered with glycans, the sugary stuff, which essentially hides the spike protein from immunoglobulins or antibodies. Right? Well, the same thing can happen with cancer cells, for example. Cancer cells covered with glycans, these complex sugars, and they prevent antibodies from attacking the glycan or at least they prevent antibodies from attacking the cancer cell because they can't see it. The cell is covered with these glycans. Okay, you following me? And there's one drug in trial right now for advanced cancer. I'm sure there'll be a whole bunch more. So really very remarkable. One other way of going at this is, a whole field of clickable antibodies. So if you develop antibodies that are specific, say, to a certain cancer, then you can infuse that into a person with one side of the click on it. And then, for example, attach a radioisotope to the other end of the click, and then the two come together, and now you know exactly where the tumor cells are in the body because they now light up. And you could use that to focus X-rays for the treatment. So it's a great idea. I mean, this is just spinning out of control. There are a lot of people involved in that now. I mean, the stuff I'm talking about now is stuff that she did in, say up to maybe 2010. So there's the gang, and that happens to be the structure of the coronavirus spikes, and the green stuff are glycans. So, and we don't need to see anything more. Wasn't that lovely, that picture of Vermeer? So, any questions on the stuff I've talked about? Because I've got, yeah.
I'm just wondering, there seems to be a lot of attention on TV through ads, especially or your doctor. They're really paying attention to blood sugars and your A1C.
Yeah, glycosylated hemoglobin. It's the glycan attached to the hemoglobin.
So is this because they're developing such knowledge of the influence of glycans?
Well, the glycosylated hemoglobin gives them an average measure over a long time. It's not like a single blood sugar level. So it gives them a pretty good idea what your average blood sugar level has been, say, over a month. That's its value.
So they're trying to keep the glycans lower in your system?
They're trying to keep the glycosylated hemoglobin down, or your sugar level.
Yeah.
Okay.
Because they see the relationship with immunity?
No, I know, that's a whole other thing.
Oh, okay.
Yeah, that's a whole other thing.
Okay.
Now, I am going to, since we do have, and with your forbearance, I am going to go into that thing on my father in chemistry. Do you mind? You do? Okay, so, well, I just put this together about 2 hours before I came here because I got thinking about my father. So my father came from a family of English Midland dyers of wool and the first to go to university. In his case, Cornell University, where he became a chemical engineer. He was a quiet, broadly read and educated man with whom I spent many summer evenings or days fishing for smallmouth bass on the upper Thames or Maitland River. Some of you may know them. He worked for a small chemical company in London, Ontario, close enough to walk home for lunch, and one night watch a frightening fire break out in the plant. I remember that. My mother got all excited about it. He said, I remember saying, Mary, that's my future. And she was enjoying this lovely fire. Anyway, throughout my childhood, he rarely talked about his work. It was only when my professor in physical, I remember this guy, my professor in physical chemistry at Western University chastised me and threatened to tell my father about my lackadaisical attitude toward organic chemistry, that I found out that my father was a member and later head of the department's monthly speakers program and much admired by the staff. Still, it wasn't until I wrote my first book in 1983 that I was forced to finally come to grips with the biophysics of electrical signaling in the central and peripheral nervous systems. That led, if you're wondering where all the articles kind of came from, the impetus, the beginnings of that, we're talking about that now. That led to learning about the genes that coded for sodium and potassium ion channels and genetics more broadly, studying proteins, the origins of life, and the atoms, which play key roles in life, such as hydrogen, oxygen, phosphorus, nitrogen, and carbon. And carbon I've likened to the matriarchal atom for its matchmaking or bond-making properties. More recently, the Nobel series helped out too, because it brought me face-to-face with Nobel Prize-winning studies in physics, chemistry, and medicine, which shaped my understanding of the basics in molecular biology, including making proteins and more recently, sugars, glycans. This year is no different. The Nobel Prize this year in medicine or physiology, remember, was accorded to Svante Pääbo. His work helped me realize how technically challenging and daunting the study of ancient DNA is, but also how much it tells us about our deep heritage, family relationships, and how small differences in genes responsible for the brain's development may be responsible for major behavioral and cognitive differences between closely related Homo species, such as Neanderthals and modern humans. The 2022 Nobel Prize in Chemistry follows earlier prizes, which illustrated how harnessing evolution can be used. Francis Arnold, I talked about. How genes may be edited, Emmanuel Carpentier and Jennifer Doudna. How enzymes can be improved, that's Benjamin List. And how genes are related to specific types of sensory receptors, that's David Julius and Arden Panaputian. This year it carries on with the Chemistry Award to Carolyn Bertuzzi, Morton Meldal, and Barry Sharpless for their studies. Well, I'm not going to go through all of that because we've reviewed it, but I'll just end up. And so on. It's all about molecules, modifying them, creating them, and using them to identify biological targets in normal cells or cancer cells. And just the last paragraph. Looking back, my father would likely be proud that I finally took chemistry seriously, and no doubt would be amazed at what chemists can do these days, and what they might do in the future. So there we are. So it's kind of a nice personal story. It didn't occur to me to think about this. I mean, it's been in the back of my head for years. We never talked about chemistry. But I have to say, chemistry's moved along a lot from those days. And he would be totally amazed. So, any thoughts? Yes?
This is a fundamental question.
Yeah.
I'm not a chemist. How do you
Neither
create,
am I.
how do you create a new molecule? And what are the basic ingredients that go into it? And once you create it, how do you know it is a new molecule?
Well, one of the things that Sharpless, I think I neglected to mention this, that Sharpless says is that, look, there isn't a great banquet of thousands of different smaller molecules. There aren't that many pieces in the jigsaw puzzle. The puzzle comes from the arrangement of the pieces in the jigsaw puzzle. I mean, remember DNA? There are only 4 bases. Yet there are, 25,000 protein encoding genes. So it's in the arrangement of the pieces. But from a chemist's point of view, there aren't that many kind of subunits or units, from which you can construct larger molecules. So that's the first thing. That's what people didn't realize for a long, until Sharpless actually said it. And that's where the field changed. And then, once you realize that there aren't thousands of subunits out there, there are maybe a few hundred, and maybe a few, and a lesser number of basic reactions, chemical reactions, well, then it all gets a lot simplified, and now, once you've got these units, all you need is something to stick them together, clip them together. That's what he found. The magic, the crown jewel, the one reaction that was secure, and it lasted, it absolutely locked them together. So now we have a way of assembling A to B to C to D and so on. And it's much easier to do. But you could imagine it's very difficult, say in the 1990s, second half of the 1990s, the 20th century, second half of the 20th century. It was really difficult. I mean, figuring out DNA in retrospect was a pretty simple thing. I mean, there are many moving parts. Proteins are a far more formidable thing. And turning out that glycans, they could have exactly the same chemistry, but different shapes. That really changes things. And remember I said shape dictates function? So glycans are kind of the chameleons of the chemical world. And I don't know, did I answer your question? I don't know whether I did.
But once you find it out, how do you know it's a new molecule?
Oh,
doesn't exist before.
they have very, very precise tools. I mean, they honed those tools in the previous 50 years, the last half of the 20th century, in their ability to identify the different units, and therefore, and in series. Yeah, so they've got the tools for that. Yeah.
It was X-ray.
X-ray crystallography
Yeah,
is certainly a tool,
Yeah.
and it's gotten a lot better. You can realize how much it got better when you look at the X-ray crystallography pictures at the time of DNA.
Yeah. My daughter was involved with X-ray crystallography of solid state chemistry. Okay, and she said the field right now was proteins. You know, she said that was taking up all the research was going to the molecules instead of the solid state, looking at reactions between.
Well, you can see why, because
exactly.
it's going places. Yeah, you don't want to be the last man using an old Model T.
Yeah.
Yeah, Jane. Oh, anything else? Okay. Well, I've enjoyed it a lot, and the three of them. So, big change next week, in the next 3 weeks. And but I put that in that thing about my father in chemistry partly because it actually pulls together the last 6 years of chemistry prizes, and you realize that there is a mind in the Nobel Committee that's working. After all, they know about what's going on. They're pretty savvy about what's going on. They can see where the trends are, and they see the nominations coming in. I think they do actually a very good job. Yeah.
Yeah, talking about the Nobel Committee, how many people are on the committee and how do they set up their committees to judge who gets a Nobel Prize each year?
Yeah, we should have a nice slide for that. For the science prizes, I can't speak for the other three. For the science prize, it actually involves several hundred people. Yeah, it used to be in the very early days, they were just Scandinavian people. And, but now they ask, well, first of all, they ask all the previous Nobel laureates in that field, where they have the option. But they also go to leading universities around the world, and people they regularly seek their opinions on. I'm thinking about Madame Curie, they were their second Nobel Prize. She won the first one with her husband and one other man. But then about a decade later, she won another one for herself. And she had lost her husband in an accident. I think carriage run over him, and brain injury. And she was on her own for a time. And then she had an affair with a married man with children, and I think he was very charming, I think. And anyway, they had this affair. And she'd been nominated, it had gone through the nominating process almost right up to the end. And there were people on the committee saying, oh, we can't do this. I mean, this would put the Nobel Committee in disrepute if we had this woman who was having an affair get a Nobel Prize. And somebody on that committee got a hold of her and said, just to let you know, kind of thing. And she didn't take it lying down. She fired off. And she said, look, this has nothing at all to do with any relationship I had. It has everything to do with the work I did. So judge me on my work, not the other stuff. The resistance dissolved, and she got it. And good for her. And she was a tough survivor. So quite a remarkable woman, I think.
So basically there's there's a real big perspective, a large amount of people involved.
Yes.
In awarding these prizes. I mean, there's the nominations and then there's everything. So it's
the nominations start in January.
Right.
For the coming year. So it's a yearly cycle. And it must be very nice to be awarded the prize, but in this honeymoon period between finding out and the big party in Sweden. And that's, the award ceremony actually takes place, I think, in December.
But I always love the interviews where, when someone's awarded one,
Yeah.
everybody says, oh, I had no idea, the telephone rang at, 5 in the morning.
Well, that's true, and that's exactly what happens.
I think is, great fun.
Yeah, now let's face it, I think it was Sharpless. No, was it Sharpless? I think Sharpless the second time. He was in a committee meeting, and no, it was Melbourne. He's in France, and he was at a committee meeting, and the phone rang, calling him out. And people do know whether they've been nominated, and they have a feeling. They go around to meetings and know people, I think, for sure. But a lot of really capable people haven't won them, and not a few adults have. But in this case, the person who he'd last been talking to had won the Nobel Prize a few years before. And while this guy was out answering the phone, he said, I'll bet that's the call. And so that's right. People waking up with their sleep to find out. Yeah, it's definitely at the top of the prizes. Is it perfect? No. Does it have flaws? It does. But no other award so far has emerged that has, at least for the science side. There isn't very much controversy on the science side. Most scientists are pretty straightforward, plain people. They're not colorful, baby. Do you ever hear of these people later? This is their moment of glory, the short thing in the New York Times, the Washington Post, that kind of thing. And they carry on, keep going. And most of them remain quite productive. And why wouldn't they? But they do like their work to be validated. I think that's important. Anyway, yeah, I think so.
Mhm.
Thank you very much. for
Okay.
all this.
So-
All right.
Okay.